Cancer's complexity can be understood through a set of distinct hallmarks. These defining traits represent the organizing principles of malignant transformation. This framework, developed by Hanahan and Weinberg, revolutionized our understanding of cancer biology and continues to guide research and treatment. In 2000, the landmark paper on The Hallmarks of Cancer was published in Cell. By 2011, the framework was updated to include emerging hallmarks and enabling characteristics. In 2022, the hallmarks were further expanded to 14, reflecting new discoveries. The concept of hallmarks has evolved significantly. Initially, in 2000, there were 6 hallmarks focused on cell autonomous traits. By 2011, this expanded to 10, including metabolism, immune evasion, inflammation, and enabling traits. In 2022, the hallmarks grew to 14, incorporating senescence, microbiomes, and epigenetics. Cancer hallmarks serve multiple purposes. They define functional capabilities acquired during tumor development, provide an organizing framework for biological processes, guide research towards fundamental mechanisms, and offer targets for therapeutic intervention strategies. Sustaining proliferative signaling is a key hallmark. Cancer cells activate growth factor receptors abnormally, continuously engage intracellular pathways, upregulate proliferation genes, and undergo uncontrolled cell division. Cancer cells evade growth suppressors by disabling key brakes on proliferation, such as TP53 and Rb. Without these guardrails, cells ignore signals that would normally halt division, allowing unchecked growth. Resisting cell death is crucial for cancer survival. Damaged cells block apoptotic pathways, accumulate mutations, and continue to grow despite cellular damage. Cancer cells enable replicative immortality by preserving chromosome ends through telomere maintenance, bypassing senescence limits, and allowing unlimited divisions, which supports tumor growth over time. Inducing angiogenesis is essential for tumor growth. Cancer cells release signals like VEGF to attract blood vessels, ensuring a sufficient supply of oxygen and nutrients. Activating invasion and metastasis allows cancer cells to spread. They break through basement membranes, enter blood and lymphatic vessels, adapt to new environments, and establish new tumors in distant tissues. Cancer cells deregulate cellular metabolism to support rapid growth. They prefer glycolysis even in the presence of oxygen, adapt to changing environments, and produce biosynthetic precursors for new cells. Avoiding immune destruction is vital for cancer progression. Tumors employ stealth mechanisms, create protective microenvironments, and inhibit T-cell activity to evade immune attacks. Tumor-promoting inflammation attracts immune cells to tumor sites, promotes growth through cytokine signaling, and paradoxically supports tumor survival rather than destruction. Genome instability and mutation drive cancer progression. With over 10,000 mutations per cell, select driver mutations provide growth advantages, and many cancers have defects in DNA repair mechanisms. Senescent cells in cancer have contradictory roles. Initially, they prevent malignant transformation, but later, their secretions promote tumor growth through the senescence-associated secretory phenotype, SASP. Unlocking phenotypic plasticity allows cancer cells to adapt. They can dedifferentiate, switch states, and evade treatment, contributing to therapy resistance. Non-mutational epigenetic reprogramming influences cancer behavior. Modifications like histone changes, DNA methylation, non-coding RNAs, and chromatin remodeling alter gene expression patterns. Polymorphic microbiomes within and around tumors affect cancer behavior. They modulate inflammation, metabolism, and treatment response in complex ways. The tumor microenvironment is complex, involving cancer-associated fibroblasts, immune cell infiltrates, endothelial cells, and the extracellular matrix. These components influence growth factors, physical structure, and treatment response. Genome instability is a key enabling characteristic in various cancers, with melanoma, lung cancer, colorectal cancer, breast cancer, and leukemia showing different levels of instability. The immune system's protective response gets hijacked by cancer, creating a pro-tumor environment. Key pathways like NF-kappa B and STAT3 become activated, driving multiple hallmark capabilities simultaneously. Chronic inflammation promotes growth factors, survival signals, and extracellular matrix modifying enzymes to tumors. Breast cancer exhibits several hallmarks, including sustained proliferation with HER2 amplification in 20% of cases. Hormone dependence is evident as ER+ tumors rely on estrogen signaling. Genomic instability is marked by BRCA1/2 mutations affecting DNA repair. Additionally, distinct subtypes such as luminal, HER2+, and triple-negative profiles are identified. Colorectal cancer progresses through distinct stages, starting from normal epithelium to polyp formation via APC mutation activating WNT signaling. Early adenoma involves KRAS mutation sustaining proliferation. Late adenoma sees p53 loss removing key growth suppressor, leading to carcinoma where additional mutations enable metastasis. Targeted therapies are designed to address specific cancer hallmarks. For sustained proliferation, drugs like erlotinib, EGFR inhibitor, are used in lung cancer. Angiogenesis is targeted by bevacizumab, anti-VEGF, in colorectal cancer. Immune evasion is countered by pembrolizumab, PD-1 inhibitor, in melanoma, and metabolism is targeted by IDH inhibitors in leukemia. Cancer cells develop resistance to therapies through various mechanisms. Target mutations alter drug binding sites, rendering treatments ineffective. Pathway bypass activates alternative signaling routes, and phenotypic switching changes cell identity, complicating treatment strategies. The cancer model faces several criticisms and limitations. Oversimplification occurs as cancer complexity exceeds discrete categories. Focus level is challenged by cell-centric views missing tissue-level dynamics. Benign overlap shows hallmarks appearing in non-malignant conditions, and linear thinking fails to capture the emergence of traits in networks, not isolation. Emerging research directions in cancer study include single-cell analysis for understanding tumor heterogeneity at unprecedented resolution. AI integration helps in identifying novel patterns across vast datasets. Liquid biopsies offer non-invasive detection of cancer hallmarks via blood testing, providing new avenues for diagnosis and monitoring. The clinical implications of hallmark research are profound. Biomarker identification guides diagnostic marker discovery. Treatment selection matches therapies to specific hallmark vulnerabilities. Combination strategies target multiple hallmarks simultaneously, and resistance monitoring tracks hallmark adaptations during treatment. Integrating new dimensions in cancer research involves shifting from an original cell-autonomous view to recognizing tumor-host interactions. The immune system and inflammation are added to the framework. A systems biology approach is adopted, understanding cancer as a complex adaptive system where hallmarks interact across scales. The hallmarks framework has transformed cancer research, creating a common language for the field. This organizing principle continues to evolve, with new discoveries refining our understanding of cancer biology. Future advances will further integrate hallmarks, guiding more precise diagnoses and targeted treatments.